All right, in chapter 2, we're going to focus in on chemical principles. So guys, chemistry is the science that studies how molecules are formed and how they interact together. And why in microbiology should we study chemistry? Well, microorganisms rely on complex chemical reactions in order to have metabolism. Metabolism is known as all the chemical reactions that an organism does. This is to help maintain internal balance within the organism. This is known as homeostasis. So this is why it's important for us to study a little bit about chemistry in order for us to understand metabolism and how organisms, as well as ourselves, maintain a type of homeostasis. So we're going to do a little review. Hopefully this is a review. It may have been a while since you've had a little bit of chemistry. However, we're going to start with the atom. The atom is the smallest unit of an element. It's going to help you form molecules. There are subatomic particles that are found in the atom. These include the electron. The electron has a negative charge. It has what we would consider a zero mass. And the reason why is they do have some mass or weight to them, but they're so small we kind of ignore that weight. These guys, when we look at electrons, these are outside of the atom's nucleus. They're going to orbit the nucleus of the atom in outer shells that we would call orbitals or energy levels. And you can see them located here in the picture. We then see the neutrons. Neutrons have a neutral charge or no charge. They also have a mass of 1, and they are located in the center part of the atom, which is called the nucleus. Do not get this confused with the nucleus of the cell. We then see the protons. The protons are the positive charged molecules. These also have a mass of 1, and these are also going to be located in the nucleus. Atoms, guys, have equal number of protons and electrons, and this is what causes them to be what we consider neutral. Okay, so if they have 10 protons, that means they have 10 positives. This will mean that they have 10 electrons, 10 negatives. So when we have 10 minus 10, it equals 0. This is what brings you to that neutral state. The atomic number is also important when we're looking at the atom. This is the number of protons that the atom has. This gives the element its uniqueness. So if you look here in this particular example, this is showing you carbon off of the periodic table. You'll see that this 6 that's located here is the atomic number. This tells you that carbon has 6 protons in it. We then see the atomic weight, or the mass, is the number of protons plus the number of neutrons. Well, we know in this case that carbon has 6 because its atomic number here is its number of protons. So we know that it has 6 protons. If we see here that the mass is 12, we can then do 12 minus 6 to find out how many neutrons are present. All right, and so this is what we're looking at for the mass or the atomic weight. Now guys, when we talk about an element are all the atoms of the same element have the same number of protons. And so when we look at the case with carbon, that means all carbons have 6 protons. If we're looking at oxygen, all oxygen have 8 protons. All right, and so this allows us to put these into a structure that we call here the periodic table. There are 92 naturally occurring elements out in the world. However, you'll notice that your periodic table has more than 92 listed. These others that are not part of the naturally occurring 92 are man-made types of elements. Now, if you look here, guys, the ones that are located in are highlighted in red, which are hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. These are very abundant in all types of cells, whether it's bacteria or all the way up to animals like ourselves. So when we look at the element, they have an electronic configuration, and in your textbook on table 2.2, you'll see that it shows you the electronic configurations of those particular elements that are very important. This includes carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. But one thing to note about these configurations with the electrons is that the first shell around the atom's nucleus, so the first orbit that's there, can hold up to 2 electrons. All right, once it has 2, then it is full, and it has to go to the next energy level, or the second shell. The second and third shells can hold up to 8 valence electrons. Now, guys, this is actually very simplified compared to what really is happening in these energy levels. But for the case of microbiology, we need to look at the fact that if the first energy level has 2, it's happy. The second and third, if they have 8, then they're happy. If an atom or an element, a full outer shell of electrons, it is considered stable. These stable types of elements don't like to react with anything else because they are happy with the number of electrons they have. They have a full outer shell. In the case here on your periodic table, you'll find these located on the very far right-hand side of the chart, and these are known as the noble gases. They contain, of course, helium, neon, argon, krypton, those types of things, but that's going to be why they are considered inert or noble gases, because they don't react with anything. So when we look here at this particular picture here, electrons fill from the inner shell to the outer shell. The first shell is happy when it has 2 electrons. The second and third are going to be happy when they have 8, or full when they have 8. So guys, if we take a look here, this is just looking at some electron configurations of some of these elements. And in the first case here, you have the hydrogen. The hydrogen has the atomic number of 1. And if you recall, the atomic number is the one that tells you how many protons that the element or atom has. And then also the atomic mass, which is the number of protons plus neutrons. Now remember also that if this is considered an atom, okay, it means that it's also neutral, meaning the protons and electrons are going to equal the same amount. So if we look here for the hydrogen, you'll see that it has 1 proton because the atomic number is 1. It'll also have 1 electron because 1 minus 1 equals the 0. You'll also see that there are 0 neutrons here. The atomic mass was 1. There's 1 proton, therefore there are no neutrons. If we look at carbon in this case, the atomic number is 6, therefore there are 6 protons. The electrons are going to also be 6 because 6 minus 6 gives us the 0, the neutral type of atom. And then the atomic mass is 12, so we do 12 minus 6 because we want to do the atomic mass minus the number of protons to get the number of neutrons, and in this case there are 6. If we move over here to nitrogen, you'll see that the atomic number is 7, therefore there are 7 protons. There's also going to be 7 electrons as well as 7 neutrons. Now if you look at oxygen, we see that the atomic number is 8, therefore there are 8 protons, 8 electrons, and 8 neutrons. Now, in most of these cases with carbon, nitrogen, and oxygen, you'll notice that all of these equal the same. The protons and neutrons are the same. The electrons, the protons, and neutrons are the same. There are some exceptions to this, and these are called isotopes. Isotopes are the same element, meaning that they have the same number of protons, because if they have a different number of protons, then they're a different element, okay? But what changes is the atomic mass is going to be different based on the number of neutrons. Neutrons are neutral, so you can have as many of them as you technically want. It just makes the element heavier. So the example here are some oxygen isotopes. This is oxygen 17 and oxygen 18. Now we know oxygen has 8 protons, so if it's oxygen 17, it has 8 protons, but it has 9 neutrons. If it's oxygen 18, of course it has to still have 8 protons, but now it has 10 neutrons. This just means the weight or mass is going to be different. However, they are still both oxygen based on the number of protons. This leads us to look at molecules versus compounds. Now guys, a molecule is formed as a result of atoms chemically reacting together to fill their outer shell. So they're going to react together to create the molecule. Now a compound, though, takes it a step further, and it says that it's a molecule that is made of different types of atoms. So let's look at the example here. The first one here is an oxygen atom. This is not a molecule or a compound because it is only one atom. In order to be a molecule, you have to have 2 or more. The next example here, though, is where there's 2 oxygens bonded together. When 2 oxygens are bonded together, this is considered an oxygen molecule. However, if oxygen is going to bond with 2 different types of atoms, we see here this is H2O, and H2O could also be called an H2O molecule, but we can also be more specific and call it an H2O compound. A molecule means that there's at least 2 atoms that are chemically bound together, but a compound has to have at least 2 different atoms. That's the key that makes these 2 things different. So compounds can be referred to as molecules, but not all molecules can be called compounds. We can't call this an oxygen compound. And the reason being is it's just made out of oxygen, therefore it can only be a molecule. All right, the next thing we want to look at are chemical bonds. Now, these are formed between atoms when they chemically react together, and they can end up completing this outer shell in order to be happy or full. Now, they can do this in two main ways. They can do ionic bonds, where they're going to actually give away or take electrons, or they can do covalent bonds, where they're going to share. There is another type of bond we're going to look at, and this is going to be a bond that forms between molecules, not between atoms, but between molecules, and that's the hydrogen bond. So let's look first at the ionic bonds. So when we look here at the ionic bonds, these are formed between atoms when they donate or give away electrons, and other ones are going to accept or take the electrons to fill their shell. One of the best examples of this is table salt. So if we look here with the sodium atom, the sodium has one lone electron in its outer shell. In order to be happy, it's going to have to get 7 more. It's going to be very hard for it to get 7 electrons. So it would be easier for it to give one away. If it gives that one away in the outer shell, the next shell is full, so it can be happy. Okay, on the other hand, chlorine has 7 electrons in its outer shell, meaning it only needs 1 more. So if it takes the 1 from sodium, it now has a full outer shell. Now, one thing to note about this, guys, is remember electrons have a charge. These electrons have a negative charge, and so if they give an electron away, the atom becomes positive. In this case, you'll see here that the sodium has a positive charge. On the other hand, if they accept or take the electron, they gained a negative, therefore they have a negative charge. Now, the reason why these bond together then is that opposites attract. So a positive charged atom and a negative charged atom will be attracted to each other, and they'll stick to each other, and this is an ionic bond. On the other hand, with covalent bonds are where they don't want to give their electrons away completely. What they want to do is share with another atom. These are going to form between atoms when they share electrons to fill the outer shell. So in this first example here, you see that a hydrogen atom has 1, and they need 2 to be happy. So these 2 hydrogen atoms get together, and they share their electrons. So these 2 electrons go around this hydrogen for a while, and then they go around the other hydrogen for a while, making sure that they're both happy at least part of the time. Okay, another example here is where we look at a methane molecule. This is where a carbon who has 4 electrons in its outer shell will share with 4 distinct hydrogens. This is going to allow the carbon to be happy, but also the hydrogens to be happy. We represent these when we write these out with a bond between the atoms as a line. These line represents a pair of shared electrons. So they are sharing those electrons in a covalent bond. Now, hydrogen bonds are going to form between molecules. This means that it's going to be a molecule of, say, like in this case, water is going to be attracted to another molecule of water. This happens a lot of times with polar covalent bonds. Now, in polar covalent bonds, it means that they're not sharing equally. I give the example of when a couple gets divorced and they have kids. The kids may spend 6 months with one parent and 6 months with the other parent. That would be considered a non-polar covalent bond. They're spending the same amount of time together. That's normally not the case. What happens is they may spend 9 months with one parent and only 3 months with the other. It's not equal, and that's what happens with a polar covalent bond. So in the case here with water, oxygen, because it's larger, it gets the electrons more often. Since it gets them more often, it has a slightly negative charge. These hydrogens, they don't have the electrons as often, therefore they have a slightly positive charge. And if you recall when we talked about ionic bonds, positive and negatives then attract. And in this case, the hydrogens, because they're slightly positive, are attracted to the other oxygens on the other water molecules. This creates those hydrogen bonds. Because hydrogen bonds are very weak when it's just one of them, but when you have lots of hydrogen bonds together, it can actually cause a very strong structure. This is why it takes a long time to heat water up to actually cause it to change from a liquid to a gas, because you have to break those hydrogen bonds. All right, this brings us to some chemical reactions. Chemical reactions involve the making and or breaking of chemical bonds. All right, so when we look here, we have reactants that go into the reaction and products that are going to come out. There are two main types of reactions that we're going to discuss. The first is synthesis reactions, and then we have decomposition reactions. So when we're looking at a synthesis reaction, this is where we take A plus B and we synthesize or make something new. And in this case, it's the AB molecule. On a decomposition reaction, this is where we're going to take a molecule, the AB molecule, and we're going to break it apart. We're going to break it back down into A plus B. In a synthesis reaction, this is known as an anabolic reaction, meaning we're building something, we're making something new, whereas a decomposition is known as a catabolic reaction because we are breaking it down. Okay, we're taking it and we're breaking it into its parts. Synthesis reactions require more energy to go into them. This is an endergonic reaction, meaning we are putting energy into the reaction. Decomposition reactions, on the other hand, are exergonic. This is where energy gets released or sent out, okay, because we're breaking the bonds, which is sending the energy out. Guys, think about it this way. If you're going to build something, let's just say you were given a bunch of blocks like as a kid and you were going to build a tower. It took work and energy that you had to put in and time to build that tower. But then if somebody comes over and just knocks it down, those blocks go flying and energy gets released, okay, and it decomposes it, it breaks it apart. Okay, so this is kind of that example. So anabolic or synthesis is where we're building something. Decomposition and catabolic means we're breaking it down. Now, metabolism is the sum of all these chemical reactions. This is going to be the synthesis reactions plus the decomposition. Every reaction in the organism gets added up, and that's what we call metabolism. So this involves both the exergonic reactions, releasing the energy, and the endergonic, where we're going to store the energy. So let's take a little closer look here at synthesis reactions. Synthesis reactions are where we're going to build new molecules. These are also again known as anabolic or dehydration reactions as well. And if you'll think about it, the word dehydration, normally that means we don't have enough water. So here in the dehydration reaction, we are going to actually remove or take out water. Some examples of these are going to be where you take monomers, which are the building blocks of something, and we're going to build a polymer. Mono means one part, polymer means many parts. So we're going to take the blocks and we're going to build something larger. An example of this is simple sugars. Simple sugars, the building blocks, of course, are monosaccharides. We can add more and more together to create complex sugars, also known as polysaccharides. Another example is where we can take the building blocks of a protein called amino acids. We put them together to make a bigger protein. We also can do nucleotides to build nucleic acids. All right, so these are just some examples of how this works. So we have the small parts added together to make the larger part, and in order to synthesize and push them together, we're going to remove water in the process. This is why it's known as a dehydration synthesis. Decomposition reactions are the opposite. These are going to be breaking down the molecules. So these are also known as hydrolysis because we're actually going to add water to break the bonds. So hydro is the water, lysis means to break. So this is where you're going to take a more complex type of chemical like polymer, mini part structure, and we're going to break it down into its monomers, its smaller parts. So this is the opposite of what we just saw. We take the complex carb or polysaccharide and we're going to break it into monosaccharides. We're going to take the protein and we're going to break it down into the amino acids. You're going to take the nucleic acid and break it down into the nucleotides. Okay, so if you look here, you have the larger molecule. We're going to add water in. This breaks the bond, which causes them to be the smaller units. There are two other types of chemical reactions. One is an exchange reaction, and this is where it's a combination of synthesis and decomposition, because we have to break A and B apart and C and D apart, and then we are going to synthesize and put them together in a different way. So in this case, you had AB plus CD. It breaks apart and rebuilds into AD plus CB. We also see that we can have reversible reactions. Reversible reactions are represented by arrows going in both directions. So this is where you have A plus B turning into AB. That's a synthesis reaction. However, we can take that AB and break it apart and make it into a decomposition reaction. So they can go back and forth constantly. Another thing we want to look at is important biological molecules. We want to look at inorganic versus organic molecules. An inorganic molecule lacks carbon bonded to hydrogen. So it could have carbon and not hydrogen, or it could have hydrogen and not carbon. All right. So when we look at inorganic, it means there's no carbon and hydrogen together. These are usually smaller molecules and they usually have ionic bonds. Now, an exception is water. It is H2O, but there is no carbon in it. And this one is going to be a covalent bond. Now, because of water's physical and chemical properties, it's able to help us sustain life. The fact that it has polar covalent bonds allows hydrogen bonding to occur. This is going to give water its properties for life. So one example is being an excellent solvent, meaning that it dissolves stuff very easily. Okay, that's a really good thing in order to sustain life. Another is that it participates in most of our chemical reactions. We saw water was part of that. We either add water in or we take water out. We see that water also has a low density as a solid, and this is why ice floats. So when we have ice that in water goes from a liquid to a solid, it is going to be less dense, meaning that it floats. Also, when we look at water, it has a high boiling point. This means it resists change to temperature. Okay, so it can maintain its temperature very well. This is what takes so much energy for you to boil that water in order to cook your noodles or your macaroni or whatever it is. All right, it takes a lot of energy. This is also why, like, if you make ice in the old ice trays, you put the water in it, and in an hour later, it's not going to be completely solid. It's going to take time to change that temperature. When we go into summer, we see this as well. If you have a pool or something like that, the pool is going to have the colder water in at the beginning, but as it is warmed up by the sun, we see that it will change temperatures, but it takes a long time to do so. All right, unless we put more energy into it by adding a heater. All right, so it resists change. And again, this is a good thing since majority of our body is made of water. We don't want to walk outside on a hot day and our water start boiling, or walk outside on a cold day and our water freeze up in our body. We want it to resist that change in temperature. Now guys, when we look at inorganic molecules, they can be classified as acids, bases, or salts. This depends on how they break apart in water. All right, so when we talked about water being able to dissolve things, that's what we're talking about here. If we put the inorganic molecule into the water and it separates into a hydrogen and whatever other molecule, this is going to be an acid. All right, so they release hydrogens. On the other hand, if we put the molecule into the water and it breaks up and gives off an OH, that's going to be a hydroxide ion, which we would see as a base. If it breaks apart and there's no H or OH, that is considered a salt. All right, it doesn't release either, so it's considered a salt. Now, pH is important because pH is going to determine how things are able to grow when we talk about microorganisms and things like that. The pH scale measures the power of the hydrogens, how much hydrogen is present there. So a solution that is below 7 means it has more hydrogens than OHs, therefore it is acidic. A solution that's above 7, okay, if we saw here it had more hydrogens, more OHs for an acid, a base is going to have more OHs and less H's. All right, and this is going to be a basic solution. If the solution has a pH of 7, it's considered neutral. Okay, so guys, when we look at the pH, as it starts to decrease, the concentration of hydrogens will increase. Now this increase is actually tenfold. So if we go from a pH solution of a 1 to a 2, it's 10 times more acidic at the 1 level than the 2. If we go from a pH of 1 to a 3, we see that the pH 1 has 10 or 100 times more hydrogens than the 3. All right, so it's a 10 time jump, not just a 1. All right, so each step is 10 away. So 1 to 2 is 10, 2 to 3 is 10, and so on. Now, one thing that's important that helps with resisting changes in this pH of going from acids to bases are called buffers. Buffers are going to help maintain homeostasis. The point of the buffer is if pH fluctuation does occur, this is going to be able to buffer it so it does not cause cellular damage. Okay, we also see that media that we grow bacteria on a lot of times contains buffers in order to give the best optimal type of environment for that bacteria to grow. Different microorganisms flourish at different pHs. Some may like a more acidic pH, whereas others may like a more basic pH. However, most bacteria have they like the common pH between 6.5 and 8.5, meaning they like it a little more neutral. Now, when we look at organic molecules have both carbon and hydrogen present. This is going to normally be based on covalent bonds as well. This allows these molecules to be actually very large, and so we call them macromolecules. These macromolecules normally have a carbon backbone, meaning they have a long carbon chain, and they will have functional groups coming off those chains. These functional groups are going to determine how the molecule will react. Does it like water? Does it not like water? Does it produce more of an acidic type of pH or a more basic type of pH? It's going to give properties to that molecule. There are 4 macromolecules that are super important, and if you'll notice these are the ones that we deal with when we talk about our diet most of the time. This is going to be carbohydrates, lipids, proteins, and nucleic acids. And when you look at these R or functional groups that are present, the carboxyl group is found mostly in your lipids and proteins, which means your fats and proteins. The amino group is found in your proteins, and the phosphate groups are found mostly in your nucleic acids and your ATP. So depending on what type of functional groups are present is going to help us determine what kind of macromolecule we have present. So let's talk about each of these macromolecules real quick. The first one we want to talk about are the carbohydrates. When we look at carbohydrates, they tend to have a structure that is 1 carbon, 2 hydrogens, and 1 oxygen. This creates a type of ratio. Okay, so it's a 1-to-1 ratio, 1 carbon to 2 hydrogen to 1 oxygen. A lot of times we represent this as CH2O in parentheses with an N on the outside. That N equals the number of carbons that are present. And so if this is 6 in this case, this would be C6H12O6. Now, when we look at carbohydrates, they have a variety of functions. When we look at monosaccharides, they're the simplest sugars. They normally have a ring of that's 3 to 7 carbons. If we look here, some examples of the hexoses are going to have 6 carbons to them. These are glucose and fructose. The glucose especially is our source of immediate energy. It's what your body prefers to use to get energy. However, fructose is a close second. Pentoses are going to have 5 carbons to them. This is ribose and deoxyribose, which are the sugars important in DNA and RNA. Disaccharides mean that there are 2 monosaccharides put together. So it's a monosaccharide plus a monosaccharide. One example here is sucrose. This is going to be found in plants. It's the type of sugar that's found in plants. And lactose is the one we find a lot of times in animals, and this is going to be found in milk products. Polysaccharides, on the other hand, are going to be multiple monosaccharides. They could actually be thousands that are put together. Examples of this are glycogen. This is how our body stores our extra sugars. Cellulose is going to make up the cell wall of most of your plant products, your plants. And starch is going to be what we find a lot of times in potatoes. It's a way that plants store their sugars. And we also see chitin or chitin that we see is going to be part of the cell wall. It's going to be part of the cell wall in fungus. So guys, when we look here on this concept, some microbes produce enzymes to hydrolyze into monomers for energy sources. So they can take these polysaccharides and disaccharides, and they have enzymes that break them into the monosaccharides, which is what they want for energy. And a lot of times these disaccharides and polysaccharides are used as structural components by the microorganism, or in order to give that organism more structure. So if we take a look here, guys, the monosaccharides, it's a single ring sugar. Disaccharide, you can see that 2 are bonded together here, which means if they're bonded together, that's a synthesis reaction, which means it's also going to be a dehydration. Water was removed. And then we also see the polysaccharide here with the many chains that are present. The next group we want to look at are the lipids. Lipids are insoluble in water, meaning they're hydrophobic. They don't like water. However, they are soluble. They do dissolve in alcohol. Simple Lipids are known as fats. Most of the time it's 3 fatty acids plus a glycerol, which is why we call them triglycerides. Triglycerides can be saturated or unsaturated. If they're saturated, this means that the carbon chain is completely surrounded by hydrogens. All right, so they're saturated with hydrogens. On the other hand, if it's unsaturated, you can see it here in the lower part. This is where there's some hydrogens missing, which causes a double bond between the carbons. Okay, so if there's a double bond present, you see that it has it's missing some hydrogens. This is an unsaturated fatty acid. Some more complex types of lipids are phospholipids. Phospholipids, if you'll recall, make up the plasma membrane of cells. We also see myclonic acid, which is going to be in the cell wall of some types of bacteria, specifically mycobacteria. When we look here at the phospholipid has a hydrophilic head, meaning it has an area that likes the water, and it has hydrophobic tails. The hydrophobic tails do not like the water, and this is where the lipid or fat is located. We also see here in this particular picture, this shows you the myclonic acid, which is located in the cell wall of certain bacteria. Another complex type of fat are steroids. Some examples of these are cholesterol and sterol. These are normally embedded within the plasma membrane to help give a type of fluidity to the plasma membrane. These steroids are found in the plasma membrane of animal cells, mycoplasma cells, as well as some fungi cells. They are a type of fused carbon ring, which gives a little more structure. Now we have proteins. Proteins are going to be built with amino acids. The building block is the amino acid. An amino acid has an amino group and a carboxyl group, and they're bound together by a carbon. The R here is the remainder part of the molecule, which is going to give us the 20 different types of amino acids. The 20 different amino acids just have a different R group. It could be simple, like you see here with glycine, where it just has an H, but it can also be very complex, like you might see here with the glutamine or the tryptophan, where there's a more complex structure that makes up that R group. But this gives us our 20 different types of amino acids. Now, in order to take these amino acids and build a protein, we have a series of steps that have to occur. The amino acids have to be put together into a polypeptide chain. These polypeptides are going to mean that there are peptide bonds between each of the amino acids. This polypeptide chain then has to go through a series of folding into a 3D structure in order to make a protein. Now, when we look at this, the way that the protein folds is going to provide the function of that protein. So the structure and function of proteins are very different based on what they need to potentially do. Some examples of proteins that are important in the body are enzymes, exotoxins, and even proteins that give more structure to the organism. So how does the protein get its shape? It has to go through a series of steps. The primary shape is seen here. It looks like a string of pearls. This is going to be that polypeptide chain. Once we have that string of pearls, we see that it's going to go into the secondary stage. In the secondary stage, it's going to create alpha helixes, so it's going to be more of a spiral, or it's going to be a beta pleated sheet. And you can see it here. It's kind of like if you were to take a paper and fold it like into a fan, or if you have pleats in like a skirt, that's what it starts to look like. We then see that it can go into phase 3, which is the tertiary phase. This is what gives it its actual 3D structure. Some proteins are functional at this point. Others have to go on to step 4, which is the quaternary. With quaternary, this means that we're going to take 2 or more tertiary proteins and we're going to put them together in order to make the protein functional. So these are the steps of the folding or building of the protein from primary to quaternary. Now, in order for the protein to work, it has to keep its 3D shape. If it can't keep its 3D shape, we see that denaturation takes place. This is where a change in the protein shape happens by breaking the bonds, and this causes the protein to be non-functional. Now, this could be permanent, where the protein can never refold and go back, or it could be reversed, where the protein could refold. There's a number of things that can affect this. Temperatures, especially high temperatures, can cause the proteins to start to unfold. Changes in the pH. If they like a certain pH, if it changes, either going high or low, it could potentially break down the protein. And also salt concentrations. These are all things that can potentially denature a protein to make it non-functional. The last type of macromolecule that we have are nucleic acids. The building blocks for the nucleic acids are the nucleotides. The nucleotides are made up of a phosphate group. You can see that here. A pentose type of sugar. This could be ribose or deoxyribose, depending on if we're looking at RNA or DNA. And a nitrogenous base. These nitrogenous bases are normally going to be C, A, T, and G. Okay, so we have our phosphate group, we have our sugar base, and we have our nitrogenous base. Now, T can also be replaced with U when we're talking about RNA. Now, purines are the adenine and the guanine, so this is going to be A and G. The pyridamines are the cytosine and the thymine, and one way to remember this is pyridamines has a Y in it, so does cytosine and thymine. Now, uracil is also a pyridamine. It does not have a Y in it, but it does take the place of thymine. So Watson and Crick were the scientists who came up with the structure of DNA in the 1950s. They are credited with DNA's physical molecular structure. DNA is going to be made of complementary base pairs, and A pairs with T and G pairs with C. This creates the structure of DNA as its double-stranded structure that's also a helix, like a twisted ladder. These hydrogen bonds are going to hold the ladder together of the two backbones. So if we really look at this, you can see it kind of looks like a ladder here. You've got the backbones located here, and they're held together by the rungs of the ladder, which are the hydrogen bonds. Also, guys, if you'll look here when we're looking at DNA, you'll see that the base pairs are A to T and G to C. When we move on to RNA, RNA is a little different. It's not a double helix, it's a single strand. Its sugar is also a little different. It's a ribose sugar instead of a deoxyribose sugar. It also has 3 of the same base pairs as DNA. It has A, G, and C. However, there is no T in RNA. The T gets replaced with U, uracil. Now, these are used a lot of times to create proteins. We see that RNA can be mRNA, which is messenger RNA. We also have ctRNA, which is transfer RNA, and rRNA, which is ribosomal RNA. These 3 strands of RNA work together in order to build proteins. This is called transcription and translation. So if we do a comparison of DNA to RNA, remember DNA is double-stranded, RNA is a single strand. DNA has the sugar deoxyribose, whereas RNA has the ribose sugar. With DNA, with its bases, it has A, T, G, and C, whereas the bases in RNA are A, U, G, and C. DNA is associated with your hereditary information. It stores your genetic information. It's the blueprints. It has the instructions. The RNA is going to be taking those instructions and actually making them into the proteins. So they are going to be the part that does the protein synthesis. Another structure that's important that is very similar to a nucleic acid is ATP, also known as adenosine triphosphate. This is a type of nucleotide. Its structure is going to have a ribose sugar with an adenine, and it's going to have 3 phosphates. This is why it's called triphosphate, because it has 3 phosphates. Now, the reason we want to talk about ATP is really important because it is going to be the principal carrier of energy. It's going to be how your cells use energy, and even bacteria and things like that. It's how we're able to then use that energy. So when we look here, ATP has got energy stored in it. That last phosphate has a high energy there. If we break that phosphate off, that's going to release the energy. And so this is going to be the currency of energy for most cells. So some molecules may have a combination of different types of organic molecules. So when we look here, bacteria cell walls could be a gram-positive. This means they have a cell wall with mostly peptidoglycan. If we break down peptidoglycan, it means it has disaccharides with polypeptide cross bridges. So it's got a little bit of a complex sugar with a complex protein. We also see gram-negative cell walls have a thinner layer of peptidoglycan. However, they have a thick lipopolysaccharide layer, meaning that it's going to have a layer that has more fat and sugars to it. This is associated with the idea of them being a little more pathogenic, and they produce things like endotoxins a lot of times with gram-negative bacteria. Now, information presented in this chapter can be applied in a number of areas as we go through this semester. The functional anatomy of cells is going to be talked about in chapter 4. Microbial metabolism, where we're going to be looking at how the cells break things down and build things up and also use ATP, is going to be in chapter 5. Microbial growth is in chapter 6. And then the control of this growth will be discussed in chapter 7. All right, so this covers our chapter 2. If you have any questions or concerns, please let us know.